Biology chapter 19 Genetic Technology Theory Video 1. So here as a basis we have to know what DNA, RNA, what their similarities and what their differences are. Then so obviously you have heard that DNA, fighting crime with DNA, is actually a part of our common knowledge now, because we can find out the relationship to a person. Whether we can find out if some person has been there in a place by using some sample of body fluids or hair or something like that. We take a sample of DNA from that and compare with the person. Then we will be able to figure out if that person has been there, or if someone has murdered somebody. Then to identify the blood. Then of course to identify whether a certain person is the son or the daughter of a man or woman is also something that we can do with DNA technology. So usually there is a process how this DNA is extracted. After this DNA is extracted, there is we break it apart using different reagents, and then we use DNA polymerase to replicate the key regions of DNA. Then we get multiple copies, and then using this we can compare it with the genetic profile. Then this comparison will lead to some sort of a identification. So that is actually a simple use of DNA and how it can be helpful for us. So now we have learned about DNA in chapter 6, the structure of DNA, and you should remember everything about it, the helix form and the pairing and all of those things you must remember. Then if you do not remember, please do a quick revision on that, then you will remember from last year. The structure of DNA was found out during the 1950s and 1960s, and we have developed from there. We have developed a lot. Now DNA technology is actually getting something of the past, because we have done a lot of things. We can scientists have been able to create clones of different species, different animals, etc. There are a lot of things that they have done. The cloning is the only reason why cloning is not a day-to-day activity is because of the ethical reasons. But they have cloned animals as part of experimentation, and because they have created these animals as part of experiments, but because there are ethical and social issues, because now if DNA from a sample of one person, if we create a clone, then there would be a problem who the who the parents are, because that person would not have parents. So there are a lot of benefits as well as problems that come along with DNA and this genetic engineering. So genetic engineering is a very useful form of science that we use in our day-to-day lives. For example, most of the vegetables or food that we eat would have at least some component of it that is genetically engineered. So in genetic engineering, the main focus is to remove a gene or genes, right? You remove a single gene or genes from one organism and transfer it to another, so that particular gene, that quality is expressed, right? So then now what happens is we take a useful gene from one organism, and then transfer it to another organism, right? So this process will now result in uh, a new formation of a nucleotide. We call that rDNA, that is recombinant DNA, right? Please don't misunderstand this to be RNA or mRNA or the natural forms, but here we are talking about genetically engineered form of DNA, that is recombinant DNA, rDNA. Now this DNA, this gene will be expressed by a organism. We call that the genetically modified organism, or GMO, or else transgenic organism. Transgenic organism or the genetically modified organism. So what we have learned so far is we can take, by genetic engineering, what we do is we take a gene from one organism and insert it to another, with the expectation that with the specific expectation that gene would be expressed. So then the gene that is introduced, the DNA that is introduced, would be known as recombinant DNA or rDNA. rDNA. Then the resulting organism which is going to express that particular quality is known as genetically modified organism, GMO, or else the transgenic organism. I hope you understood. So then this genetic engineering will give us solutions for many different problems that we have been having. One such problem is the removal of the barriers to gene transfer between species. So now, for example, a gene that we have, we cannot transfer it to another species unless we have sexual interactions with that, and those sexual interactions also should combine with that species, because we cannot have interspecies combination of these our gametes. We cannot do that. So what we can do is a useful gene of a different organism without any sexual activities, we can get it transferred to any other animal or person or plant or whatever it is. So then if we take even a completely different domain or a kingdom. Now for example, a bacterial gene, we are very much different to bacteria, but bacterial gene can be inserted into a plant or even a human being, and also we can do the reverse. Human gene that produces insulin is given to bacteria, so they produce human insulin. So like that, we carry out different formats of genetic transfers. As a result, we have lot of potential in genetic engineering, right? So this is not like selective breeding. In selective breeding, what we do is we use the whole set of genes. An animal who is more suitable for that environment, we select them and then we breed them. But this is completely different. We take only a part of the DNA, that is a single gene or several other genes, and add it to another person. Usually it is a single gene that we transfer. So this we have to go through we have to go through a huge process in here. Now let's look at this overview of gene transfer. Right, a GMO, that is a genetically modified organism, can be produced in many different ways, but there are some steps which are going to be essential. The first one is we have to identify the gene that is going to be transferred. Right, then what we do is it will be cut off from the chromosome, or using mRNA we can make it by reverse transcription, or we can synthesize it from nucleotides. Then what we do is we use the polymerase chain reaction. You don't actually have to know great details about how the process is carried out, but you need to remember the steps. Then what we do is we use a technique called polymerase chain reaction, PCR, and that will produce the multiple copies of the gene. Then what we do is we insert this gene into a vector which would deliver the gene to the cells of the organism that we are going to manipulate. The vectors could be plasmids, viruses, or liposomes. Then the vector will take the gene into the cell. So these steps are very, very important. There might be other steps also, because this is not as easy as it easy explained here. There are steps, more steps, but these steps are essential. Then the cell will take in this gene, and then what we do is we identify this gene by using marker genes. There are some special kind of genes that we can identify their nucleotides with. We identify these new cells and then we clone them. Right? So that that is the process. So you clone them and you can actually enhance the number of cells. So genetic engineer would need a certain set of tools, right? So the tools are not just like a mechanical engineer would need his wrenches and vice and all of those things. A genetic engineer would also need some tools. So these tools would include enzymes such as restriction endonucleases, then DNA, ligases, and reverse transcriptase. So those are the enzymes that he would need. Then he would need vectors, of course, to transfer the gene to the cells, plasmids and viruses. Then then genes genes coding for easily identifiable substances. So the gene markers are also required. So these three things are going to be the essential parts of the toolkit that should be had by the genetic engineer. So these are the keywords that you need to know. Genetic engineering. So that is the process by which a GMO is produced by transferring a single gene from one organism to another. Right? So then it is the organism that was introduced with the new gene would be known as GMO, genetically modified organism. Then recombinant DNA, that is the DNA made by artificially joining together pieces of DNA from two or more different species. Then transgenic organism, whatever the organism that contains DNA from another source other than the natural form. This DNA could be from another organism of the same species or a completely different species. Then GMO, we discussed about it. So GMO is any organism that has its DNA changed in a way that does not occur naturally. So the vector is a way of delivering genes into a cell. Used in gene technology, we use plasmids and viruses to carry out this process. I hope you understood. Let's write down a note, and after that, let's go to the next lesson. Write down. There are many methods by which genes can be introduced to one species, to one organism from another. This transfer of genes can take place between organisms of the same species or organisms from even two different kingdoms. Leave a line, put a star and write down. In genetic engineering, a gene is removed from one organism and transferred into the genes of another organism in such a way that its phenotype is expressed. I am sure you remember what phenotype is. That is the result of the gene, or the quality that is expressed as a result of the expression of that gene. The DNA that accepts the gene from another organism would be known as recombinant DNA. Recombinant DNA, within brackets, simple R, capital DNA. And the organism which shows or expresses the new gene would be known as transgenic organism or genetically modified organism, within brackets, GMO. Then write down an overview of gene transfer. When producing a GMO, following steps are essential. When producing GMO, following steps are essential. Number 1, identifying the required gene. And cutting it off from the chromosome or producing it using mRNA, within brackets, reverse transcription, or synthesizing it using nucleotides. Number 2, producing multiple copies of the gene using polymerase chain reaction, PCR. Number 3, inserting the gene using a vector to deliver the gene to the cells of the organism. The vector could be a plasmid, virus, or a liposome. Then right, number 4, the vector takes the gene into the cells. Number 5, using a marker gene, the new gene is identified and would be cloned. Leave a line and write. Genetic engineer would require following instruments within brackets tools to carry out this process. Number 1, enzymes such as restriction endonucleases, DNA ligases, and reverse transcriptase. Number 2, vectors such as plasmids and viruses. Number 3, gene markers to identify the changed genes. If you understood, please do question number 1, which is going to be with a reference to earlier lessons that you have learned. We will discuss tools for the gene technologies in the next video.